Hello and welcome to Teach Me in 10. Today we're speaking with Zehra Nizami from PartitionBio, winners of the ELRIG Innovation Award. In this conversation, we'll explore the technology behind their award-winning platform, the challenges it aims to solve in drug discovery and bioscience research, and why she believes it has the potential to unlock capabilities that weren't previously possible. Hi, Zehra. Thanks so much for joining us to speak more about your ELRIG Award and what it means for PartitionBio.
Well, thanks very much. It's a real pleasure to speak with you and to get this another opportunity to talk about this fantastic award.
So, for those unfamiliar, what is the ELRIG Innovation Award and what does it aim to celebrate and what did it mean for you to win it?
The ELRIG Innovation Award is open to all those promising startups who have won a place in the breakthrough zone. It's yet another example of ELRIG's amazing support for the drug discovery community. In the breakthrough zone, ELRIG spotlights startups that are generating disruptive new technologies and giving them an opportunity to exhibit in a portion of the whole that experiences really brilliant footfall and engagement, thanks to the thought that the organizers have put into the conference meeting and the planning and the way that everyone interacts with each other. The breakthrough zone is really hopping. Participants there can also put their hat in the ring for the overall innovation award with a short video pitch. And for us, when we won, it was a tremendous platform because we got to present our technology on the main stage to all the drug discovery participants for that year. And our booth was immediately super active afterwards. And we had a great time sort of having a little celebrity moment. We also felt that the boost that comes with the external validation of getting out there for the first time with our tech and with our big ideas. And it was nice to get the judges' perspective on that. And also the footfall that felt after that would suggest that other people agreed that this was pretty exciting.
That sounds brilliant. So, PartitionBio has won for innovation. What's the core innovation your company's developed?
Well, we won the ELRIG Innovation Prize for developing and launching a new class of scalable cell delivery technology, which we have commercialized for researchers as BubbleFect. We've named this in this way because our technology allows customers to spontaneously generate liquid-filled droplets or bubbles containing their biologic cargoes of interest. The major problem that our technology addresses is the increasingly acute challenge of delivering designer drugs into cells because in the modern era of precision medicine, these increasingly tend towards being large biologic molecules like DNA and RNA and gene editing complexes like CRISPR RNPs. And these can't naturally cross the cell membrane the way a small molecule drug can do. And while there are historically solutions to this problem, those include like mechanical disruption methods such as electroporation or viral delivery methods, or even chemical delivery methods like lipid nanoparticles. None of these can actually traverse all the various limitations that occur when you're delivering drugs at this kind of scale, such as scalability or formulation complexity or cargo size. So like how big your nucleic acid cargo can be, and the cargo type limitations. You really have to have charged cargo. So we with BubbleFect have developed a completely novel class of cell delivery that's also scalable because it's non-viral and it's non-lipid, and it's based on the principles of disordered domains that occur in proteins that are able to undergo liquid-liquid phase separation, which is like a wordy thing, but it's basically this idea that you can form these droplets containing your cargoes. And at PartitionBio, we've developed libraries of synthetic peptides that are short enough to make these kinds of molecules at scale, but also specialized enough to encourage this phase separation in the presence of a universal range of biologic cargoes. So this therefore brings us to how the platform is really particularly innovative. It takes us beyond what is realistically possible before. See, the spontaneous self-assembly of our delivery vehicles means that we can not only phase separate and generate droplets containing DNA and RNA, which are like your bread and butter cargoes, but also enables delivery of uncharged cargoes that are very underexplored, like proteins, and complexes of cargoes such as CRISPR RNPs, which was only really feasible before with mechanical disruption methods. And those ones are not really scalable or really have a track towards in vivo. So we think that this is why ELRIG sort of picked us out, because it sort of addresses current methods that are in use, but also unlocks future cargoes that could be in play for people in developing precision medicine cures.
It sounds absolutely fantastic and like it's going to make a huge impact on the community. So, looking ahead, where do you think the biggest impact of PartitionBio will be in the next few years?
Well, yeah, thanks for that feedback. And yes, looking ahead, we're hoping that PartitionBio will be able to impact in at least three different areas. Firstly, we want to successfully tap into and support the existing research use only transfection market. So wherever there are pain points, like people having trouble with a particular cargo type, their cargo is too large, like it's a giant, maybe even a human chromosome, something so big, something so difficult to imagine, or in a sensitive cell type, because that's another thing, the balance of you're putting in something really novel, not expected to the cell, and there's a lot of it, and then the cells are unhappy to take it up. And so these sensitive cell types are enabled by our technology because the liquid droplets are quite natural, and it's part of a natural uptake process that happens with the cells, known as macropinocytosis. So it's a really natural process. And then we also hope that by just sharing, and ELRIG really helped us as a sort of launch our platform by sharing and widely disseminating our technology, which is very easily to disseminate because it's room temperature stable. We hope that people will understand and appreciate the novelty of our technology and start using it widely. And then secondly, we hope that we want to nurture and develop the partnerships that we get out of the best use cases that are coming back. So once people take up our technology, we can hear where is it best applied, what cell type, what cargo type, what application, what problem, and to work together along with our internal R&D and move into in vivo applications. So this is this sort of transition from being research use only to like maybe we can start talk about gene delivery and gene therapies and things like that. And then thirdly, we also push our own internal R&D towards enabling step changes in the current problem. So even within transfection, we've identified that one of the problems is how do you do delivery of cargoes in non-dividing cells into the nucleus of the cell. So you need to get DNA and gene editing complexes into the nucleus. When cells aren't dividing, the nuclear envelope is a bit of another barrier. And our technology, because it enables putting in loads of complex cargoes into it, you can make a more complicated paradigm to sort of bypass the nucleus, and we're working on that. So we think that our technology sort of enables step changes. So these are the three main big areas we want to move into. And we've already sort of started that progress with, and again, we got a great launch at ELRIG in doing so.
Brilliant. Well, thank you so much for joining us to speak about that. And congratulations on the win, and looking forward to seeing more innovations from PartitionBio.
Thanks, Laura, and thanks to ELRIG.
Thank you. Bye bye. Thank you to Zehra for joining us today and for sharing the story behind PartitionBio's award-winning technology. ELRIG is a not-for-profit organization built on the idea of community, offering open access and free-to-attend events for the global life science and drug discovery industry. Be sure to look up their next event, and we'll catch you on the next episode. Bye for now.